And we're going to open up with the poll. Oh, okay. So the first question is, what type of treatment are you currently taking? Hey, Gina, if you can keep an eye on the waiting room, that'd be great.
Okay, I definitely can if I'm not on camera. So I'll try. I gotcha.
All right. Okay, I'm going to end this poll. I'm going to share the results. And since we're recording, I'm going to announce out loud what medication you're currently taking. Alectinib is number one at 63%. Lorlatinib is at 12%. And then we have 7% of chemotherapy and 7% of brigatinib. I'm going to stop sharing that, and I'm going to go to our next polling question. How long since your diagnosis?
Mark, is that one working? Because I don't see it on my screen, but I'm on an iPad, which is a little bit weird for tonight.
It should be working because the votes are coming in.
I got
great.
it.
Okay, good.
Okay, so 37% 1 to 2 years, 27% 1 year and under, and then 3 to 4 years is 10%, 5 to 6, 8%. We have actually 7 and 8, which is 18%. So that kind of goes hand in hand with the alectinib. And I'm going to go to the next question. This is how many TKAs have you taken? And you also can indicate if you've taken chemotherapy as part of your treatments. This is, you have 2 choices if you want. All right, and the poll. And share the results with you. Can you see that? Can everyone see that? Nod your head. Okay.
Yeah.
So we have 1 TKA is 53%, 2 TKAs are 20%, 31% of us that have taken chemotherapy, and we've had a few 3%. Excuse me, 3 TKIs at 14%. So another question coming up is, this is an easy one. I'm assuming, what's your stage of diagnosis? Okay, I'm going to end this one. Okay, 83% of us are at stage 4. Okay. Next question. When you have progressed, what area was your progression? Okay, I'm going to end the poll. So this one we have 20% was in the lung, 53% of us have not had any progression. And then we have a variety of bone at 14%, actually, and brain, excuse me, at 14%, and bone is at 10%. And then I think I have one more question. Okay, hold on. Oh. Okay, this is my, this question came up because I want to have you guys realize what you're missing if you don't come June 1st to our party of our ALK-positive party, where we have some funny questions that we ask and funny discussions. So vote away. I'm curious to see how many drunken sailors we have out there. Gina's cracking up because she can't believe I'm doing this. This was a part of our pregame.
This was not part of our pregame, but it's awesome.
All right.
So
we have a lot of Bahama Mamas at 68% and some screwdrivers at 22%. Well, anyways, I'm going to turn it over to Gina. And we have two wonderful doctors to speak with us. I just want to let you know that this is number 15 and 16 of the doctors so far in our yearly ALK Talks. So we're so appreciative of these doctors spending their Sundays with us. And I don't realize, you guys have to realize how fortunate that we are, that we're speaking with world-class doctors. For some reason, they're all at MGH. Maybe I should move to Boston. I don't know. But anyways, Gina, take it away.
Oh yeah, I like that shirt you got on there, Mark. It's looking good. He's got his Walk Positive shirt on. Walk Positive challenge is going on all through the month of May. So if you guys want to, it is not too late to sign up for the Walk Positive challenge, where you take 10,000 steps in 10 days or $10 to donate towards the positive. We are so excited that you guys are here. You're doing an amazing job raising some money for research, just like Dr. Iafrate and Dr. Bar-Peled. And we are just so happy that we have you guys here. And we're very fortunate to be able to have a team of medical professionals, ALK-positive patients, key opinion leaders, all working together to pick the most impactful research. And some of that research that we picked was that of Dr. Iafrate. And you'll find soon that that's because he has been in this space for a long time in the ALK space, and he's been doing a lot of great stuff. So I wanted to introduce you to two of our medical committee members. They are amazing to volunteer their time and energy to ALK positive to make sure that we're looking at the very, very best research. So I wanted to introduce you to a friend of mine, Jenny Blenders, and Ricardo Salgado. If you guys will just both take yourselves off mute, and I'll let you guys say hi to everybody, and then I'll give the meeting over to you guys.
Hello.
Hey, Jenny, you want to go?
All right, sure. Hi, everyone who doesn't know me. I'm Jenny Blender. I was diagnosed ALK positive in 2015 and doing great still. I've been on the medical committee, I think pretty much since it's been formed. And in my non-cancer life, I'm a clinical psychologist. And I'm glad to be here tonight as part of this wonderful talk tonight.
Thank you.
Ricardo?
And I'm Ricardo Salgado, and I'm one of the new kids on the block. I was diagnosed on October 15th of 2000, so less than 6 months ago. And I learned that I was ALK positive on October 29th of 2000, and it was like the best day of my life. And literally the next day, the first thing I did was call Gina. And the next day I was listening into the ALK positive summit. And there was so much wealth of information that I was able to consume in that weekend of really identifying not only the top, doctors, but the top research. And Dr. Iafrate, who was one of them that had been awarded. And, it really accelerated my whole journey of understanding. And it's been such a phenomenal resource and privilege that I've been able to really get up to speed in such a short period of time. So I'm very grateful for not only the organization, but the doctors like Dr. Iafrate, who spend their lives, sacrificing their personal life to push the boundaries of medicine to try to save ours. So with that, I'll turn it back. Gina?
I can share my screen with you so you can start your presentation, Doctor.
Okay. You see that okay?
Yep.
Can you hear me okay?
Yep.
All right. Thank you so much for the invitation. Of course, also thank you for the research support, which is incredibly valuable to my group and my colleague, Dr. Liron Bar-Peled, who's here with me. The two of us are going to do a tag team presentation. Also, I believe I have one of my lab members here, Diane Yang, who's actually doing the research work, who's with us, and maybe I can convince her to show her face and tell you a little bit about what we're doing in the laboratory. I think the way we'll do it, I'll give a little bit of historical background about how I got involved in ALK, what we've done, where we stand with the disease, and what are we think some of the really interesting research directions that we can go in. And then Dr. Bar-Peled will take over. He's a chemist also at MGH. He's a chemical biologist and is an expert in a very fascinating new area of drug development. And I've been very fortunate to get to know him and work with him and propose the project that we proposed for this research. So you'll get to hear a little bit more detail about chemistry as well and drug development. And then we have some kind of preliminary data from this project. We've been doing it for about 6 months, so we've made a little bit of progress, and hopefully Diane can give you a little bit of oversight of that. So let's see if I can move forward here. Gina, Mark, if it's okay, I'll give a little bit of historical overview. They said I was involved from the beginning. That was really true. I started at MGH out of my training. I'm a pathologist, and as a pathologist, I focus on diagnostics. So probably of the people you've invited, I would be the one to ask about diagnostic tests, if people have questions about that. That's my area of expertise. This is a summary of lung cancer. The genetics of lung cancer around 2017 is still about true. When I started in 2005, EGFR mutations were just described. So it was 2004, right before I started. And I was really excited when I started my job at MGH that we were entering a new world of personalized medicine, especially in lung cancer, for which there really was nothing. Personalized medicine was a hope for the future. It wasn't reality. And that the group we have on the call today is here to talk with me is just it's just amazing how much progress we've made since then. So EGFR is over here. It's, as some of you probably know, it's about a quarter to a fifth in the Western populations of lung cancer patients. And then ALK, as you probably very well know, is around 3% of all lung adenocarcinomas. So it's a small slice of the pie, but a very important one, and one that I believe strongly, and I have since I started, that you have to take care of even patients with even the rarest subtypes of tumors, and that's a critical area of focus. And I think we have done as a field. We're not perfect, and we haven't cured the disease, but we made a lot of progress, not only against ALK, but ROS, RET in the last years, MET exon 14. There are NTRK fusions that aren't listed here that are like even less than 1% of tumors. So it's been a really rewarding time to be involved in personalized medicine. ALK, as you know, really was started by the Japanese group of Dr. Hiroyuki Mano in 2007. That was a landmark paper. It was very interesting for me scientifically for a few reasons. One, it was a new important mutation in lung cancer, one that we could think about targeting. But it's a gene fusion, and you probably know a lot about gene fusions now. Most of you probably never heard of gene fusion before. But it's different than the more common types of alterations in cancer, which are mutations, like single point mutations, like you might see in some of the resistance mutations, or gene amplifications, like a HER2 amplified breast cancer. Fusions really weren't known outside of rare subtypes of tumors like lymphomas, leukemias, and sarcomas. And this was one of the first ones described in what we call epithelial tumors. And it opened up a whole new world of fusion research, which is what my career has been based on. So I've really focused first on ALK and then other fusions. Dr. Mano's paper, of course, described the fusion between EML4 and ALK. The fusions are fair, in some ways very straightforward to understand. They take the enzymatic activity of ALK, which is the kinase activity, and fuse it to a part of this other gene, EML4. These two genes don't go together normally. You probably have read all about this. These genes aren't normally fused together. This is like a Frankenstein, where two halves come together. Why this happens? many of you probably have asked yourself, why did this happen to me? Why did this happen in my cells? We don't know. Unlike some forms of cancer, like smoking-related cancers or melanoma in sun-exposed individuals, we don't know what causes these gene fusions to happen. We know that they almost certainly involve the chromosomes breaking and then being rearranged and coming back together in a strange or unusual way. But we don't know why. I have an interest in research, which is separate from the work that we're going to talk about today, on what could cause these type of rearrangements and other specific signatures. Each of you on the call has your own EMO4 ALK or other ALK fusion. Maybe some of you don't have EMO4, maybe a variant fusion. But the ALK part of the fusion is almost all the same. So this is one common theme with the ALK fusions and the ROS and RET fusions as well. It's almost always the same part of ALK, and that is the C terminus or the 3' end, which is the kinase domain, the enzymatic activity. Usually the breaks are around exon 20. Don't need to go into too much detail about exon numbering, but we have different flavors of EMO4. They all have something in common. You may ask, what is it about EMO4 that is so magical, that makes ALK go from basically a quiet gene not causing cancer to suddenly being cancer causing? It does two things. First, ALK is not expressed in the normal adult lung. So there's no ALK just floating around in your lung. None of the cells lining your alveoli or in your bronchi express ALK. And so EMO4 is a gene that you take the front part of it and put it next to ALK. It now makes ALK expressed because you kind of put them together. You now drive ALK being expressed. That protein is now present in those cells where it normally isn't. And the other thing is EMO4 does something called dimerization. Dimerization is when two proteins stick together, and when two proteins coming together you'll hear or read about this all the time in cell growth signals. How cells divide and grow often involves two proteins coming together. That signal coming together turns it on. So it's critical to dimerize, turn on ALK kinase activity, and that kinase then signals the cells essentially to divide. So that's what EMO4 is doing. EMO4 usually doesn't cause cancer. It's not really associated with cancer. There are some forms of cancer that are associated with ALK mutations. We won't talk about that, or ALK amplifications. But the fusions are specific almost entirely to lung cancer. We have ALK fusions, obviously, in other tumor types. So how did I get involved? I'm a pathologist. I actually ran the molecular, in some ways still work in the molecular diagnostics group at the Mass General. And after Dr. Mano's paper came out, we very quickly set up a test to detect ALK rearrangements. Some of you may know there are ALK rearranged lymphomas, anaplastic large cell lymphoma, where ALK gets its name. And we had genetic tests at that time, way back in 2007, I guess, the end of 2007, to detect the lymphomas. So we engineered the same type of test. What I'm showing here is something called a FISH, fluorescence in situ hybridization test. And we had that available and set up. And it was really a perfect storm, so to speak, in late 2007, because we had a trial. Well, I'll come to that in a minute. We had a trial of a drug that was going to work in patients open already. We didn't know ALK fusions existed at the time. That was the amazing thing. We had the trial open, but we didn't know anything about ALK fusions. So it all came together in a perfect storm at the end of 2007, the beginning of 2008. Maybe I'll go forward one or two slides and then come back. Yeah, so this was the trial that was open. This is the drug name, PF something, 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 1066. It's amazing to me that many of the folks on this call never have heard of this drug, but probably are never going to get it. This is crizotinib. So this is the drug that became known as crizotinib. This was the first drug approved in ALK-positive patients. Many of you are never going to get it, which to me is amazing. And so we had a FISH assay set up, And we had an outstanding hematologist, oncologist at MGH at that time, a physician called Tom Lynch. That name rings a bell. Those of you who read about personalized medicine might know Tom was the first author on the EGFR mutation paper. And Tom was a great partner. Myself in the laboratory, he as an oncologist seeing patients. And we set up, essentially started setting up a connection where we would start running the FISH assay on some of his patients. What was interesting at the time is what became Presonub was open under Jeff Clark. I don't know if anyone here knows who Jeff Clark is. Jeff Clark was the principal investigator on the crizotinib phase 1 study. And he and I also were very fortunate to work together. This trial was set up as a MET inhibitor trial. So we were looking for alterations in the gene called MET. Some of you may have heard that. There's evidence of MET as a resistance mechanism in ALK-positive patients. You can develop MET alterations later on. There's also the more famous MET exon 14 skipping lung cancer. That's an important part. And so we were studying this gene. It's also a kinase. It's very similar to ALK in what it does. And we had this trial open looking for MET-positive stomach cancers. Actually, it was not even lung cancer. It was a stomach cancer trial. And how to say this, the trial wasn't going well. We were having a very difficult time finding patients. Part of running these trials, especially in rare subtypes, are finding rare patients. Now with ALK positive, patients can find you, right? Patients will identify themselves as ALK positive. At the time, that wasn't the case. We didn't genotype everyone. We didn't know who was ALK positive. We didn't know who was MET positive. We had to actively look at them. So we screened all of our stomach cancer patients for MET. We didn't find any. And so Pfizer, along with Jeff, were considering closing down this trial. And then Dr. Mano's paper came out. And it was very interesting because it was a MET inhibitor in its early days. That's how it started its life. And deep in the small writing of the clinical protocol, it said, well, there's some off-target effects also. And one of the the targets not of crizotinib that we're not interested in, but we have to pay attention to is this other gene called ALK. No one really cares about it, but you should be aware of it. And so Jeff was very smart when Dr. Mano's paper came out and said, well, instead of killing the trial, why don't we turn it into an ALK trial if we can identify those patients? And so that was the perfect storm. End of 2007. That trial was open at MGH, phase 1 trial. We set up a test, and by the beginning of 2008, we identified the first ALK-positive patient to go on to a TKI. And I would say that was probably one of the most rewarding periods of my career. We were able to find a relatively large number of patients in that first few months, and many of them had really remarkable responses. Mark did a survey where he showed how many of you presented with stage 4 disease. So, pre-TKI, pre-tyrosine kinase, ALK-positive tumors are an aggressive disease. Many EGFR patients present at low stage. They can be managed surgically. But ALK presented with metastatic disease at stage 4 in most patients. And that's still true, as you saw on the survey. So we identified using FISH at the time a number of patients. I remember the first 2 patients. Maybe you're interested in hearing a little bit of their stories. I can't say their names, but their stories. The first one, imagine this being the first one that you identify it. We're pretty sure we're right. We're not totally sure, but we're pretty sure we're right. We know this is ALK positive. The patient, Pfizer said, no, we will not put you onto this trial because we didn't have enough information. There weren't even animal models. There weren't really good cell models. Like, we were just taking a leap of faith that we should try this for a very sick patient. This patient self-advocated with one of our senators in Massachusetts to call Pfizer to allow them to be put on this. And again, I can't use any names, but they self-advocated. They ended up going on drug and had a bad reaction to it, which was very unfortunate. And we were nervous that this drug was going to have severe side effects that would limit our ability to make any progress. It was an unfortunate first patient. The second patient, I would say the first patient, this was in January of 2008. The first patient, although he had side effects, had stage 4 disease, was on 2 liters of oxygen, and considering what to do next in terms of, you can imagine what the discussions were. And he was skiing within a month after that. And we were all just blown away. And Tom Lynch, again, was really critical to making this happen, along with Jeff Clark and Eunice Kwok. Eunice ended up being the first author on the original crizotinib paper in the NEJM. So quite a whirlwind of activity. So we got the first patients on there. That this was amazing in a few ways. One, when you try a new drug in a population and you see an amazing response, that drug now became standard of care at MGH, right? This was the first research patient we tried. And now every oncologist at MGH had to have ALK fusion data on every patient. Right? This was not a phase 3 trial of 500 people with 2 or 3 years of survival. This was one patient, one genotype. Now we have to do everybody. And that was one of the magical parts for me of being at MGH. We had great leadership who were really open to doing this. That mantle was obviously handed to Alice Shaw in short time. And Alice, you know the story behind Alice, did a phenomenal job. And many of you are going to, have a lot. We all owe a lot to Alice and what she's done for the field. But that's a little bit of the earlier story. So 2009, we put together the first data set describing the clinical parameters. Dr. Mano's paper really was the science behind it. It wasn't talking about the clinical parameters, but he talked about the clinical parameters of patients. We had patients here. This is showing the average age of patients with ALK versus non-ALK. Many of you on the call probably were diagnosed at a young age. The median age for lung cancer is probably 65, 70. Many of the ALK-positive patients the average was around 50, and we had patients in their 30s. Some of you I can see are young on the call, and that was really different. That was much different than EGFR. And it was a little shocking at the time. And that statistically is very, very significant. Smoking history, we also realized that relatively early, and we published this in this paper with Alice, that it was independent of smoking. I don't like to call this a non-smoker's disease because that implies smoking protects you against it. Right? Do you understand what I'm saying? So if you say it's only a disease of non-smokers, that means, oh, smokers don't get it. Smoking must be good to prevent against ALK. So I'd like to maybe mark a question of how many people were smokers. I would bet about 20% because it's independent of smoking. We don't think smoking has anything to do with how these things happen. It should be independent of smoking history. And so the same number of people walking down the street that smoke, right, that percentage should be the same percentage of people who are ALK positive smoke. And that's in the US, it's about 20%. So my bet, Mark, would be if you ask that question, we'd have 20% smokers or current or former smokers on the call.
I may have to add that to my list of questions. Maybe I'll knock the drink question off. My guess is you're right. I have not met many ALK smokers.
Yeah.
Maybe we should have smoked a little in our life.
People smoke less, so the amount of smoking comes down. And then you can see this very striking link to stage 4 disease. We saw mostly stage 4 patients. So we described that. I'm a pathologist. I don't know if there are any visual people on the call, but the ALK tumors look different. They have a very unique picture under the microscope, and that's my training is in looking under the microscope at things. And this is what a tumor might look like under the microscope. It's very specific appearance, to the point where it's about half the, I don't want to say 50% of the time I'm right. about half of ALK tumors I can just pick out by looking at the microscope. I just be like, that's an ALK tumor, and I'm almost sure. And this is one that's a classic appearance of an ALK-positive tumor. We also described the use of diagnostics of fish. This was the first paper describing the use of immunohistochemistry to diagnose ALK. I imagine some of you on the call were diagnosed with immunohistochemistry as the main test. So we described 2 of the first diagnostic tests. And then one of my postdocs described the use of next generation sequencing, a sequencing panel that we developed in the lab called anchored multiplex PCR, which is now one of the common assays used for fusion detection. It's a specific assay we designed for fusion detection. So as I said, my career has really been finding diagnostic approaches to fusion detection with a large amount of work on ALK. We talked about Presonub a little bit. To have the first drug in class have a response that looks like this. You've seen these kind of what's called waterfall plots, where each bar is a single patient, and this is the size of the tumor based on radiology if they increase or decrease on drug. And this looked like EGFR mutant lung cancer on EGFR inhibitors. Really, really impressive. And so the 2007 to 2010 time was incredibly exciting for ALK patients. It really changed everyone's perspective. Together with Dr. Shaw, we began to study patients who progressed on crizotinib. Many of you may know that you had a question on where did you relapse. The majority of patients in those days relapse in the brain because crizotinib is not brain penetrant. So there are a lot of failures in the brain. Alectinib kind of ended that problem, which is one of the reasons alectinib is such a great drug. It gets into the brain and prevents brain mets from growing. Presonub had that problem. So we had patients who relapsed in the brain without any mutations, just because the drug didn't get in there. We identified some of the common mutations early on. So we described the first description of mutations in ALK, as well as what's called, for those of you who are scientifically inclined, bypass tracks, other signaling pathways that get activated in the resistant patients. So we described all of that. I'm going to skip over a whole bunch of the middle part of this story and leap to the end, which, as many of you know, there have been multiple drugs developed that address the common mechanisms of resistance. Lorlatinib being the current, I would call it state-of-the-art, or the big gun that you have, for many of you will have left. Lorlatinib works against gatekeeper and solvent front mutations. However, we do see resistance to lorlatinib, and this paper from our MGH group also shows that some combinations of mutations still mediate resistance. So for that reason, Liron and I began talking about putting together a proposal, developing drugs that would work against some of these resistance mutations that even lorlatinib won't work against, and considering approaches that would develop drugs against EML4 ALK using different mechanisms of action. So most, or all, I should say, of the approved drugs right now target the kinase domain itself, the enzyme portion of the protein that, let's just call it the active part of the protein. And the drugs bind here with these PSAR, those are phosphates that are essentially the currency of a kinase. So they block this phosphate groups from being added, and that blocks ALK from being active and essentially kills the tumor cells. But the tumors eventually evolve resistance by mutating those sites. And so our project, and I'll hand it over to Liron now, is addressing the need for drugs with novel mechanisms of action. And with some of the technology Liron has developed, I got super excited that we have a different approach. So could you develop drugs, for instance, I mentioned this need for two proteins to come together, right? That's the EML4 part of the protein. You really need that to happen. What if you could prevent those two things from coming together? Like get in the middle of those two EML4s and block them from coming together. So you can block dimerization. You could induce degradation. So proteins are folded in very specific, highly organized manner, right? The proteins are long amino acid stretches, but they have to fold into a three-dimensional structure. some drugs, and Liron is an expert in this, some drugs can get into a protein, like throwing a wrench into an engine, and you can cause the protein to unfold, to unwind a bit, or become unstable. And that's not necessarily specifically focused on the kinase domain itself or the phosphate active. So degrading the fusion protein overall or getting local misfolding of this kinase domain. And those were our approaches, and we thought, based on some of Liron's prior data, that we'd have a decent shot of developing new drugs with that. So with that background, I'll stop sharing and hand it over to Liron.
Dr. Iafrate, before we switch over, can you answer a couple of the questions that people had about what you were presenting so far?
Sure.
So the first question was, is there any significance in the relative locations of the mutations you showed around the pie chart?
The first pie chart, the big pie chart.
Yeah.
No, it's basically based on how common they are. So there's nothing magical about them in terms of how bad they are, when they happen, the age, or anything like that. They're just listed in order of most common to least common, or in some way, in a way that's easy to look at. But no, there's no meaning to the location of the fusion on the pie chart, or the location of the mutation on the pie chart.
Great. And just a definition, what does variant mean when you use that term?
Variant, yeah, great. So our genome is made of 3 billion bases, right? And those bases are A, C, Gs, and Ts. And the amazing genome consists of 3 billion A, C, Gs, or Ts. The sequence, I haven't memorized the genome, but it could look like A, C, G, T, G, T, C, G, T, G, 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 something like that. It just goes on for 3 billion base pairs, packaged into different chromosomes, right? So we have 22 chromosomes and an X and Y. And so those 3 billion base pairs are packaged into volumes, book volumes, like an encyclopedia. What is a variant? A variant is a change to the sequence of that book. So imagine, if any of you remember encyclopedias, imagine the Encyclopedia Britannica, all the volumes, change one of the letters in one of the volumes, just one letter to a different letter, that's a variant. And that's because we have the Encyclopedia Britannica as the printed version. You bought one with a mistake. And one of the most amazing things about molecular diagnostics and tumor genetics is oftentimes we're looking for one change to the letter code, a single letter change in the entire Encyclopedia Britannica, right? One letter change can cause a tumor. In the case of EML4-ALK, I guess if people will bear with me on this analogy, which I like, it would be like splitting 2 volumes in half and then stitching them together, right? So you had like two volumes that didn't come together, and now you open the page and you're like, oh, that's half of one page and half of the other. What's going on here? But a variant can be, we usually think of variants as a single base change, but it could be a fusion or an amplification. It's anything that's different. It's a variation from the normal sequence of the human genome.
Great, thank you for that. So we also got some excitement about Presonub, and for you for being part of this from the beginning. Some gratitude, it looks like. Another question, how fast does ALK spread? Since so many are diagnosed at stage 4, is it a matter of weeks to months from stage 1 to 4, or what are we looking at?
Yeah, it's a good question, and no answer. I don't have an answer, so I usually have answers. I don't have an answer. There are mathematicians who work in biology, and they can model how big a tumor needs to grow before you see it, how many cells you need to see it, and then based on what we know of the machinery that copies DNA, we can predict how many mutations would you expect, let's say in 10 billion cells. Let's say your tumor, tumors can have billions and billions of cells in them. How many mutations or variants would you have in that? So we have people who study the changes of DNA over time. What we don't have a good handle on is the natural history. Why would a 1 centimeter EGFR mutant tumor and a 1 centimeter ALK tumor likely behave differently, and that is you're likely not to catch that ALK tumor when it's just 1 centimeter and only one little nodule in the lung. We don't catch them. And I don't have an answer why we don't do that. It's a simple question, but it's a biology that we don't understand. But the obvious answer is EMO4 ALK creates cells that not only can grow in an in a non-controlled proliferative manner, that is, they can divide and divide and divide without stopping, but they also have the ability to travel into the bloodstream or the lymphatics of the body and travel elsewhere and find homes elsewhere in the brain or in other parts of the lung. And what's the biology behind that? We don't have a really good understanding of why that is, and why perhaps the fusion itself is such a powerful, what we call oncogene. It's a very powerful gene that can get the cell to grow and spread almost by itself. Now, that's the downside. The good side of having what looks like one really powerful, we call them drivers. They drive the tumor, the driver genetic alterations. The good part of that is you really only might need one drug, because most other tumors need multiple genes mutated and altered at the same time to get a fully malignant tumor that spreads. Here, it's really one alteration that seems like enough. And so it's bad news in the untreated setting where you're presenting with high stage tumor, but your tumor has an Achilles heel that other tumors don't, and it has one of them. And so if you just can really focus on that one, do a great job with good drugs, you can have a good outcome. We're hoping to continue to work on that paradigm, that if we just hit ALK better and harder from the start, outcomes will continue to improve.
All right, fingers crossed for that and all your good work. One more, it looks like someone noted that there's a low incidence of Asian ALK patients on the graph. Wondering if you have any insight into that.
I don't have a lot of insight. There are a lot of Asians in the EGFR population for reasons we don't fully understand. Obviously, a lot of ALK patients in Asia, but I don't think we see the same enrichment in Asian patients that we see in EGFR. Where really there's a remarkable incidence in Asian females. So no, we don't fully understand. One of the reasons MGH got the lead in this field, you may be wondering why there's so much MGH people. I showed you this graph that described the age and the stage, that table. In that table is the number of ALK patients. In that first cohort of patients we had, 13% of MGH patients had ALK in that first year or two study that we did. And why was that? Why that was much higher than anyone else had seen, right? 3% is about right. We had so many patients that let us study them, and it let us run the trials. So we had this kind of head start on everyone that really was a feed forward cycle. And then of course we had Alice Shaw, and she's just a force of nature. But why do we have so many? We had so many because Tom Lynch was at MGH, and EGFR mutations were discovered by him and Daniel Haber. And that was a disease of non-smokers. Again, I'm sorry, I use non-smokers there loosely. And so we had many patients coming to MGH to see Tom because they had lung cancer and were not smokers. So it's like maybe outpatients coming to see certain specialists now. We had a lot of non-smokers coming to MGH. And the ones who were EGFR negative had a pretty good shot of having ALK. And so we had an enrichment. All right, I'm sorry, I'm taking up too much time, and Liron needs to describe some science.
No, it's good. It's good. It's good to hear.
Well, we have a couple more, but we can save them for after, it looks like.
Yeah.
Do you want to go ahead, Dr. Bar-Peled?
I sure can. I'm going to share my screen. Boom, boom. And then, can you guys see the title screen? And John, please help me out if I get a little too chemistry heavy here. Want to make sure I'm not as eloquent as John, as this will become quite apparent. But yeah, so it's really quite an honor to be able to speak to both ALK positive and the patients with ALK. And what I thought I would do today is tell you about some of the technology that we're developing in our lab at MGH in collaboration with John, and how we're hoping to use this new revolutionary platform to eventually develop drugs targeting ALK and ALK-driven non-small cell lung cancers. So, I think it's very, very fair to say that cancer genomics has really revolutionized how we understand cancers and the genes which drive cancers. And since 2000, thanks in large part to advances in DNA sequencing technology, we've identified something close to 700 genes that when mutated or associated with cancer. Roughly 400 of these genes are bona fide cancer drivers and are required for the proliferation of cancers. And from these genetic data, we've actually been able to make some really transformative medicines. So of these 400, roughly 42 have been targeted successfully. And these include some very notable genes which mutated, such as EGFR, BRAF, and ALK should also be on here, but I didn't include it. However, what's quite disappointing is that while we're able to target 42 of these cancer drivers, if you do the simple division, we see that we're only targeting roughly 10% of bona fide cancer drivers, right? So that means that the remaining 90% of genes that cause cancer are completely untouched by pharmaceutical companies or by the best drugs that we have today. So why is that the case? Well, it turns out that the genome, that only a very small portion of the genome is druggable, or in other words, encodes for proteins that can be attacked by drugs. So common genes which are considered druggable are kinases, and in particular kinase domains, right? So ALK is a kinase, and it contains, the way it's folded means that you can develop a small molecule for it, because people have developed many drugs against different kinases. However, the vast majority of the genome, as you can see here in black, encodes for genes which are undruggable. And it turns out that roughly 80 to 90% of cancer drivers fall into this undruggable space. And so while a lot of big pharma companies traditionally work in this much more druggable domain, we think that there's really a tremendous untapped opportunity by focusing on the undruggable space. And again, by undruggable, it doesn't necessarily mean that we don't go after kinases. We just think of certain non-druggable domains, right? So as John alluded to, if we can block EML for dimerization, that wouldn't necessarily be by targeting this much more druggable part of ALK, but rather what would traditionally be considered undruggable. And so really the question is, how do we access this undruggable space? And it's really more of changing kind of the perception in the field about what is druggable. And essentially, we're just changing what people think, right? So our solution is to simply expand the druggable proteome. And the way we do this is by targeting one particular part of a protein or a gene known as the amino acid cysteine. And if there's one thing that you take out of my little talk here, is that cysteines really are important for the function of proteins. They play critical roles in protein functions. And I'm sorry, this is a little bit more jargony. For example, they're required for the way that proteins function. Some proteins function. They hold the structure of the protein together. They tell where the protein where to go within a particular cancer cell. And they also allow for certain proteins to come together. And it's really the unique chemistry of cysteines which allow it to function as sort of a handle to attach drugs, or a new class of drugs for this. So targeting cysteines is actually very much established. So there are very good drugs right now, such as osimertinib for EGFR, that actually targets a cysteine within the EGFR kinase domain. Similarly, for B-cell malignancies, another drug, ibrutinib, targets another critical cysteine within this oncogene. Now, to kind of give you the scope of targeting cysteines, in the human genome or in the human proteome, there's something like 250,000 cysteines. Now, by our most conservative estimates, we think that roughly 10% of all of these cysteines are highly druggable. And so if you simply do the math, you multiply 250,000 by 10%, and it's probably going to be more like 20%, but we're quite conservative. That'll mean that there's roughly 25,000 druggable cysteines encoded in a cancer cell line. And so currently, we're roughly at 25%. The best estimate, 25% of the human genome is druggable. And we hope that in the near future, by using these technologies, we'll be able to have something like a 5 to 10-fold increase in the number of druggable proteins. And so that will essentially mean we're going from 25% to something closer to 90%. And that's really, really good, because that means that more of these potent oncogenes or potentially mechanisms of escape from traditional TKIs will now be targetable using this approach. So the question is, how do we identify all of these newly druggable cysteines, right? And the answer is something that we call chemical proteomics, and it's a very new technology. It hasn't been around for more than 5 years, so everything that I'm telling you about is completely cutting edge. So chemical proteomics, kind of as the name suggests, is a combination of chemistry, very, very specific, we call them chemical probes, that bind to certain cysteines within the proteome. And in order to identify which cysteines these so-called chemical probes bind, we use a very powerful instrument called a mass spectrometer. And essentially, what a mass spectrometer does is it simply reads or it sequences all of the different proteins that this particular chemical probe binds to. And so essentially, what this allows us to do is to characterize protein druggability on a global scale, on an entire, for an entire cancer cell line at once. Traditionally, and I should have put a slide of this, but the traditional approach that many different large pharmaceutical companies use is to go after their favorite protein, to devote all their energy into their favorite protein and figure out whether it's druggable or not. We're much more, how do I put it, opportunistic, in the sense that we view the entire totality of druggability within a cell, and then we figure out which protein to go after. And usually we love to be able to correlate that with that protein's importance in driving the proliferation of that cancer. So I thought, oh, I'm a bit stuck here. Whoops, no, I'm going back. Sorry about this. All right, so I thought I would kind of give you a high-level overview about how our technology works. So when we first start any one of these projects, the most important thing for us is to make a cysteine druggability map, right? The cysteine is a particular part of that protein that we want to target. And what we do is we start with a cancer cell line that we might have growing in the lab, or now we've actually been able to transition this to patient samples. So when a patient gets operated on, when their tumor is removed in the OR, we can actually now make a map of all the druggable proteins. And the way this works, and again, John, you might want to help here with the jargon. What we do is we crush or we open up a cell, and then we treat that cell with these chemicals, these compounds called drug probes. And what these drug probes do is they tell us when they bind to a particular protein, a particular cysteine, is they predict which proteins are druggable. And we use that information to immediately hone in on the proteins that we want to go after. So after we do very complicated bioinformatic analysis, we identify all the druggable proteins. And then comes a very challenging part. We have to develop small molecule inhibitors or drugs against these different druggable proteins. And the way we do that, and hopefully Diane will be able to tell you about that, is we build very specific biochemical or cellular readouts for the function of a protein. And then we screen our lab's library of roughly 4,000 different cysteine specific drugs. And so when these drugs inhibit, or when these drugs bind to a particular protein that's druggable, they block their function. Now, the uniqueness of our approach is that in addition to doing this more traditional biochemical screen, we do this secondary proteomic screen. Now, this is really, really important for the following reason. We know that we want to not only measure the engagement or the binding of our drug to the target that we want to see, but we also want to measure the off targets, the other proteins that these drugs bind to. And that's really, really critical. John told you a really great story about how crizotinib came around. And that was because it was being designed for a different kinase, but one of the off targets happened to be ALK. Much the same way, we want to be able to use proteomics to find all the off targets. And this is really important, not only to serendipitously be able to drug other types of proteins, but also during the drug development process, we want to make sure that our drug is really targeting what we think it's targeting. And that will reduce the toxicity that's associated with a lot of older versions, older drugs. And from there, and I won't get too much into this, we do more traditional chemistry to take some of the hits from our drug screen and develop them into real drugs, which will eventually go into patients.
Liron, could I stop you for a second?
Sure.
I have a question just with this slide itself. So you're talking about individual ALK tumor from a patient, and then you go ahead with your analysis. So your CDM workflow chart that you're showing us, this is not for an individual ALK patient. This is for an overall ALK EML tumor. Is this what you're showing us? You can do a new lorlatinib type of a molecule to develop? Is that what you're showing us here?
Right. So our work right now, and the funded work, which we're extremely grateful for, centers on starting with cell lines, which were eventually, which were once came from a patient. What I wanted to indicate is that in the next generation of this approach, we should be able to take a patient's tumor and then identify all the different types of druggable proteins there. In other words, let the patient's tumor tell us what we should be drugging.
So this is the future, is what you're showing us.
Well,
not
this
like
is
today.
This is both today and in the very, very near future.
So if you biopsy my tumor, you can go ahead and crush it, put the analysis in, and you're talking about this is a 14-day map. 34-day map.
Well,
I mean, is that what
it's
you're
2 days.
trying
2
to
days
do?
it's 2 days to get the map.
Yeah.
It takes the drug development, drug discovery process. Unfortunately, takes a little bit longer. To do the initial screen is very, very quick once we have a very robust way to look at the function of a protein. What unfortunately takes a long time is moving that hit compound from something that works on cells to something in animals to something that's safe and tolerable for patients.
So
John,
do you currently have an ALK patient that's doing this?
That is something that we're very much interested in beginning. Initially we thought that we would start with bona fide human cell lines or ALK-positive cell lines, make a map there, and then immediately or in the very near future extend to patient samples.
Okay, so that makes sense to me. So this is in the future something that you would love to do, where we individualize to each patient's tumor.
Yes,
I would,
exactly.
Mark, I would maybe give a little bit of a different perspective. I don't think we would ever consider using this as a clinical diagnostic assay. That is, we could take your tumor, figure out which drugs work against it, and then give you those drugs. There are approaches like this. This is really to come up with a list. On the far right, you see lead compound. You'd want to come up with a list of drugs or chemicals right? We're starting with thousands of chemicals. We want to identify the handful of chemicals that will target our genes or proteins of interest. Those will all be different drugs, different structures, and all of them will then have to be put into animals. They'll have to be optimized for all the usual drug type of things, PKs, pharmacokinetics, distribution, toxicity, all the usual drug development things then have to go on. So I think Liron's describing a very short time frame to do the research assay, but the full drug development is, a 2 to 3 year time frame.
Right, because it's going to have to go through the phase of the trials,
Yeah.
etc.
2 to 3 years is a
Right.
short time frame for.
So are you currently using this model to come up with other things besides lorlatinib at this point, currently?
That's exactly the approach. And Liron's ability to drug the undruggable now opens up EML4 dimerization. He has a number of other hypotheses. Well, I'll let him finish. I'm sorry.
Yeah, no, that's a great segue. So, in our previous work, we had taken a non-small cell lung cancer cell line, and we simply made one of these maps. And what we discovered is that, very excitingly, EML4, a cysteine, this residue, this critical residue within EML4, which is, again, required to bring these two proteins together, is very, very druggable. And that's really exciting for us because it suggests that we can find a new approach to target EML4 or these ALK fusions that's not targeting the much more druggable domain, this kinase domain, where a lot of the resistance mechanisms happen. I think what's also very, very exciting for us is that within this EML4 or this ALK signaling pathway, we identified additional proteins, they're sort of lieutenants of ALK, which are required in driving the cancer. And we found that there are many of these proteins which also contain these druggable cysteines, and we think that they're druggable, that we would be able to develop small molecule inhibitors or chemicals to block their activity as well.
Gotcha. So those are the SHP2 inhibitors?
No, did I show that here? No. Well, for example, I show here that we identified a potentially druggable cysteine on STAT3, which is important in the activity of ALK in promoting this uncontrolled growth.
Yeah, I'm just trying to explain the graph to dumb it down for some of us, including myself. But Jenny and Ricardo, I don't know from this graph if you can maybe ask some questions or dig a little deeper into some of this discussion.
I had a quick question. How does this overlap with protein degraders? I mean, I know here you're trying to intercept before the proteins bind together, right? Before that happens, but is it different?
Yes and no. So some of the current protein degraders, which are in clinical trials, are being developed for. They require, unfortunately, two components of a molecule. One of the components brings this degrader. It's called a ubiquitin ligase, which degrades a protein. The other part of that small molecule has to bind to a protein, and that's really the problem. That second small molecule that has to bind to a protein, that's the problem, because again, it's still based on the idea that you can only target certain classes of druggable proteins. So we're kind of throwing that entire idea out. We think that if we can target any cysteine or the cysteine that we identify as druggable, we can target them. And so that kind of short circuits the requirement of these degrader molecules, because again, the degrader molecules will only target those traditionally druggable proteins. It's a new way of targeting them, but they're still very much focused on the more traditionally druggable proteins, if that makes sense.
No, that's super helpful. And I think Mark was going into this, but he was trying to get a little bit better clarity on timeline and status and all this. And for me, it wasn't really clear, but would love clarity on, like, right now, the status of this project is, obviously, we're not trying this yet on humans or onto the cell line, side, but any like best guess of like the timeline from here when it could enter the clinic?
Liron, why don't we finish up with the slides, Ricardo, and then we come back to
Go
that.
for it. Yeah, go for it.
Yeah, so I'm done with kind of the overview, but if Diana's on the line, maybe she can show some of.
I have one of her slides, Liron, if you can
Stop share.
turn it back over. Diane, are you on the call?
Yes.
I don't know if you're comfortable, but this is all Diane's work from this project. She's made a lot of progress. So maybe if you can just simply take the team through what one of these experiments looks like.
Okay. Can I share my screen? I actually had a little bit. Okay.
Yeah. I added this part to the slide, Diane, so just so everyone knows, Diane's already finished screening 5,000 compounds. So that part of the project has already been completed. And those libraries are libraries of different chemicals that are related in that they bind cysteine, but they all have different structures.
These compounds are FDA approved compounds?
No. These are new, these would be new chemicals. To become an FDA-approved compound needs to go through the next steps of drug development.
Gotcha.
Drug development.
Okay, so I think I will just use a few minutes to update what we have been done in the lab. And first, I will start with explaining how the screening was done. So we took the cell line, which is the EML4 ALK version 1. Variant 1's positive cell line, the HT122, and we treat them with either the DMO. So here is used as a solvent treatment control, and the positive control we use is lorlatinib. And we use the multiplex library, which we combine 10 compound and mix together into a single wells of cells. And we have screened total of 4,000 compounds. So in this assay here that you've seen is called the Western blot. It is a method to quantify the protein amount. So what you are seeing here is we are looking at the levels of the phosphorylated ALK, which is known to be an active form of ALK. So what you can see here is the lorlatinib treatment significantly decreased the phosphorylation, which is the activations of the ALK. So the compound that we are looking for will be similar to the actions of lorlatinib, but it is a cysteine reactive compound. So what we have found so far, so this is just an example of it, but we have identified around 20 different pools of compound mix. So for example, the number 7 here is the 10 compound mix pool that shows the decrease of phospho-ALK, which is the activations of ALK. So this is what we decided to follow up. In the next stage of the screen, showing at the example here, I actually just got this data on Friday, so really excited. So we expand this 10 compound mix into individual compound. And again, I guess it's just a lucky 7. So we also found the compound number 7 also significantly decreased the ALK activity. So now compound like this further we will further validate it and then enter the third stage of the screen. And we will further repeat and validate and find out what this compound actually hit in the molecular target. So this is the summaries about what we have so far. And we total identified 19 of this compound pool, and we're gonna expand each of them and then to test whether we can find more reactive cysteine compound that will decrease ALK activity.
Mm-hmm. Yeah, so maybe to clarify a little bit, I think between the three of us, we didn't make it clear. So the project does two things in parallel. One is it does the cysteine mapping that Liron mentioned, which is, Liron essentially has developed. And that is, we're in the middle of that. And that's we have that data, but it's not fully analyzed, where it gives us a list of cysteines, of all the proteins in the genome. We know in these cell lines, we know which cysteines in which proteins are what Liron calls druggable, where a cysteine reactive drug could bind there. And that's an important list because it tells us of the important important targets, bypass targets, or ALK itself, where can we possibly bind a drug? In parallel, we're actually doing the cysteine screen. So with those two things in parallel, starting with ALK as the obvious target, of course, we would want to know which of these drugs bind or inhibit ALK activity. So those two experiments go in parallel. This mapping gives us all protein information, not just ALK. It tells us about all the proteins in that cell that are potentially druggable. And so that's if the following phase will be to follow up on that large data set and potentially find new proteins that could be inhibited in combination, let's say, with an ALK inhibitor, or something along those lines. So we kind of simplified the story, but there's multiple experiments go on simultaneously. But I would say I'm super happy with the progress. Diane has done an amazing job so far in going through this. So thank you, Diane.
I'll second that. Yeah, it's really, I mean, we're just chugging along really at a very fast pace here.
It's really exciting. I think we should go through some questions really quickly here. Jen, you want to start reading them?
Yes, I'm just trying to see where we left off. Thank you so much for that. It's very exciting. We were finishing off a little bit ago talking about few Asians in your sample. Someone else was asking about why is it enriched for women?
Yeah, I think a lot of the original data set that we showed was very biased. So I said we had 13% ALK positive. We know that's not the general population. That was our MGH special population in the beginning. So I think over time we've learned more, and the sex preference, I don't think there's a strong sex preference for ALK. So take some of that early data with a grain of salt. Some of it ended up being incredibly important, right? That they're younger patients and non-smokers. That was like the big take home from there. So that's what I would suggest you digest and take from those early studies.
So I think I'm going to switch back and forth between both of you with questions. You were talking earlier about the morphology. Is the distinctive morphology of the ALK tumors specific to adenocarcinoma ALK, or is it also true of squamous cell?
Now, we don't see a lot of ALK in squamous cell carcinoma. Are there people on the call who've had squamous cell carcinoma?
I know we have had a few cases or some that have transformed. But I mean, I don't know very many people.
Yeah, so these are two morphologically distinct types of lung cancer: adenocarcinoma, which grows as round glands, and squamous cell, which looks like skin under the microscope. It's this layered epithelium that looks like skin. It's squamous. I've only seen a few true squamous cell carcinomas ALK positive over the years. If anyone on the call has that, I'd be very interested in hearing a little bit more of your history. Maybe it's not best to do that in public. One patient that I remember distinctly who had a true squamous cell at diagnosis had a history of radiation exposure. And that patient had a prior breast cancer with radiation to the chest wall. And I've always been convinced that it was radiation exposure that gave rise to a second malignancy. And that's an area I mentioned. I'm interested in what causes these kind of tumors. One thing I'm very interested in is exposure to radiation, whether it's prior medical radiation or radon exposure or anything else like that. So very interested, especially in patients who had squamous at diagnosis. If any of them are on the call, I'd be interested in hearing if you have any past history. Again, it's pretty rare.
I have a general question, and that is, how do you share information with like Dr. Kalari and Dr. Awad and others in the that are trying to develop a vaccination, ALK vaccination?
It's interesting. We have there's several mechanisms. Those at MGH, we obviously have close collaborations with all of our MGH colleagues, and we work together on these projects. We are submitting together with Dr. Awad across town. He's on the other side of Boston. He trained in the system in Boston, which means he rotated at all the institutions. So I know him well when he was a trainee. He's fantastic. We've submitted to the NIH a combined lung cancer research program called a SPORE. SPORE program is the acronym for this specialized research group, which includes Mark and our team at MGH. So we're hoping if that comes together, that it'll be a much more solid collaboration throughout the Boston groups. But for now, we do have a number of mechanisms to collaborate and share. And as you might know, all clinical trial protocols that are open at the Dana-Farber are open at MGH also. We're actually within a single administrative unit.
Have you looked at the TPX0131 molecule?
We've not used that molecule in our lines yet. I think that's something in terms of, we didn't really talk about what do we do next to follow up on hits. Mm-hmm. But the validation of hits includes comparing the activity to other existing drugs, to compare it to see whether these hits will inhibit mutated forms of ALK. Of course, we'd have to do the resistance mutations. Are they inhibited? If they are, that's our main goal, right? Can we develop drugs that work against the resistance forms? Mm-hmm. And one of the other magical things that Liron's capable of doing using his mass spectrometry is to actually know which drugs, where in all the proteins do the individual hits bind. He'll actually tell us, like Diane said, number 7 in pool 7 look great. Liron will tell us in a few months, these are the locations in the genome, or in the proteome. The proteome, the definition of the proteome is all proteins. So we have about 20,000 proteins. In which of those 20,000 are the drugs binding? And we expect them, Liron can correct me, to probably bind somewhere between 10 and 50 proteins. These compounds we're talking about now, which are the research compounds. He'll be able to tell us which proteins are bound. And then it's our job as lab scientists to go through those lists and figure out which one of those hits, those targets that the drug binds to. We're hoping some of them are ALK, EMO4 ALK, but they may not be. They might be other targets. And we need to Diane will be sorting out more work for Diane. She'll be sorting out which one of those targets are the important one. And that will help us in the future drug development phase. So a lot of work to be done to validate the responses, including looking at other drugs, which you mentioned, looking at resistance mutations, and then mapping the actual binding site. So a lot of work to be done.
Looks like we have a few more questions, and Ricardo, feel free to jump in rather than me monopolizing. How can you tell an ALK adenocarcinoma from a non-ALK adenocarcinoma by looking at the morphology, and how exactly is it different?
I don't think a lot of pathologists use that information, but having looked at a lot of them, I think I showed you a picture. Maybe I can share my screen again if those of you are interested. Let me see if I can bring it up. Yeah, the ALK tumors often are what we call solid. Instead of forming little circles, they form like one clump. Like all the tumor cells look like a clump. And within the clump, they have these forms called signet ring cells. A signet ring, you may know what a signet ring is. It's essentially like a ring. So if you think of an engagement ring or a signet ring. And it's this special shape where the cell is round, but the nucleus is pushed to the side. Most cells are like a fried egg. They're round with the nucleus in the middle, right? The nucleus is where the DNA is. It's like a fried egg. These look like fried eggs where the yolk is pushed to the side. And the shape ends up being a ring, like an engagement ring, where the nucleus looks like the diamond. And so this picture of a solid tumor with lots of signet ring cells floating in the middle, to me, is just ALK. And not all of them have this picture, but I would say, in my experience, about half of them. I'm a pathologist, so I talk about this all the time.
I do have a few questions on the, you mentioned, some of the most interesting times of your career were when the Presonub or what, those like casual encounters or discoveries. And when Dr. Amano's paper came out, how intensive is that collaboration, with like the Japanese or Asian community to collaborate? And I mean, you guys run such an amazing center at MGH, but there's so much other stuff going on abroad. Is that like really, is there a tight collaboration? Would love any insight on that collaboration.
Yeah, I would say not as tight as it could be. I mean, we have great colleagues. Maybe it's too long of a conversation, but Liron and I and all researchers know about collaboration versus competition in science.
Right.
And oftentimes people who support research want everyone to collaborate. This is my personal opinion, by the way. I wouldn't say a general opinion of scientists. I think it's okay that people compete as much as they collaborate. You want some collaboration. You don't want repeating science. You don't want incremental science. You really want to move things forward. But being in Boston, and Liron can shake his head up and down if he agrees, being in Boston, competition drives a lot of progress. And so I'm always balancing in my head, especially when I'm asked about, why don't you collaborate more? Well, sometimes competition makes you move fast. So I think we could collaborate more. We've had some large national collaborations on personalized medicine, on lung cancer mutation consortium I was involved in the beginning, was many sites across the country. I was still am involved in something called the NCI MATCH trial, which is a very large multi-center trial for personalized medicine. So we do a lot of collaborative work, mostly towards the back end, the clinical trial development side, to be honest. The earlier stages, it tends to be competitive, and that includes with Asia. So depending on which stage you're talking about in terms of development, the earlier stages tend to be more competitive, where you don't see a ton of collaboration. And often I work with several foundations. The foundations, really want people to work together, right? They don't want to invest their money to just have those tools limited here. How come you're not helping those people there? They might move faster if you gave them information. So I think it works well, and I think foundations are doing something good by encouraging collaboration. Funding research is a good way to encourage collaboration, to be honest. Because if there's money available only in the setting of collaboration, people collaborate.
Awesome. And then the last question for me is, if putting your pathology hat on, is there anything for our community that you would advise us that, you might think it's standard of care to MGH, but it's not maybe standard of care across, like I'm still surprised that only 20% of non-small cell lung cancer get next generation sequencing. That for me is like shocking to me when I was diagnosed. Is there anything else beyond, like, obviously get your next generation sequencing, but like, a liquid biopsy or anything like that our community should hear that is best practice? Or if you were, even better, if you were in these shoes, this is what you would be doing?
Yeah, I mean, I think the good news is the field, in many ways driven by Alice, has done an amazing job, and the current guidelines are pretty mature now, and we have good drugs where people can stay on for a few years and stay stable. I think once you've relapsed, then it's a different question, where you probably want to seek out a center of excellence where they're doing things like cell-free DNA, they're doing monitoring tests, genetic monitoring tests, and they have new cutting-edge trials. That may seem obvious to you, and maybe seem obvious to everyone on the call, but I can only imagine. I've had family members with cancer. I haven't had cancer, but you're dealing with a lot of complicated issues, including how far are you willing to travel, where are you willing to go, what are you willing to do. So I think finding a center of excellence that you can at least get an opinion on is, it's an obvious statement, but I think that's important. It's really hard to say there's anything standard of care right now that, like with the original crizotinib, like if you don't do this, you're missing out. I don't think that exists right now because the field has done an amazing job.
Thank you.
It's kind of a follow up to what your last statement was, and that is 2012 when I was diagnosed, the life expectancy has pushed the envelope now, and when you're diagnosed now in 2021, for the old timers in our 12 or 15% of the people in this call, what do you see a future drug coming down that will be a game changer or something that could help us?
I mean, let's say you relapse on lorlatinib. The question will be, what's the mechanism of relapse? If it's multiple mutations, right now we don't have an answer for that. But if it's bypass tracks, I think you have multiple options, which include combinations with certain types of chemotherapy, combinations with other tyrosine kinase inhibitors, MET inhibitors, EGFR inhibitors. I think there's all kinds of really hopeful combinations that haven't been needed so far now. Well, they've been needed, but we haven't had a rational approach to them. I think you'll begin to see more combinations that can be used in the resistance setting, especially post lorlatinib. So a lot of people on the call I see haven't had lorlatinib yet, which is great. That means that they still have kind of the big gun that can work against many of the other resistance mutations. So I think we'll have a lot of combinations coming out, and I think there are a lot of the drug companies will begin to see the benefit of extending the life of patients in your setting even further by having those. And it's great. I mean, it's fantastic that we're talking about that. I mean, it's such great news.
Yeah, that's great news. I don't know, Jenny or Ricardo, if you have anything else. I know we kind of ran past our time.
Yeah, there's still a few more questions, but I know we're really pushing it on the time. So.
Well, if you want to grab one or two, we have John and Liron, unless they want to bounce off the call.
Yeah, I would say
questions.
I would say to our team, to the ALK-positive team, you can forward us questions by email, and I'm happy to answer them directly to anyone who has them. Even if they don't have them now, if you have a question later on, I'm more than happy. I'm sure Liron's more than happy to discuss chemistry.
Can you type your email into the chat?
Yep.
And a phone number?
I'll give you Liron's phone number.
Right.
Cell phone.
Yeah, cell phone is I really appreciate the time that you put into it and your heart and soul's in it. And you saw it from the very beginning, which is amazing, the transformation that's happening. And like you said, we have an Achilles heel, but we have one Achilles heel. I think that's how you put it, right?
Mm-hmm.
Yeah. As many of us know on the call, having lung cancer, ALK lung cancer it's kind of like winning the golden ticket. You're not smiling.
It's still cancer.
Yeah, it's still cancer.
You got half a golden ticket.
Yeah.
Well, it was our pleasure, and to thank the group and everyone who's done fundraising. I know you guys put your heart and soul into the fundraising effort, and we take very seriously doing good things with that money. Right? It's either your own personal hard-earned money or your friends or family, and so we just say we do our best to do well by your hard work and dedication.
And thank you so much for showing up, and you guys can unmute yourself and thank the doctors for their participation. Jenny and Ricardo, thank you so much.
Thank you.
Thank
guys.
you.
Thank you.
Thank you.
Thank
Thank
you
you.
very much.
Thank
Thank you so much.
Thank you.
Thank you.
Thank you all.
Thank you.